Good day. My name is David Monson, Senior Director of Business Development at Salonic. Welcome to today's presentation. During the session, your microphone will be on mute, though we invite you to ask questions as they arise using the chat and question tool on the right-hand side of your toolbar. We'll ask your questions to Sebastian following his presentation. At the end of today's webinar, you'll receive a brief survey. Please complete it so we may continue to provide you quality presentations in the future. Thank you. Today's webinar, sponsored by Salonic Group, is titled, Difficult to Express Proteins? Choose the right human cell line with a proven track record in large-scale GMP perfusion production. I'd like to welcome and introduce you to our presenter, Dr. Sebastian Lalouvet, Team Manager of Cell Line Development at Salonic. Good day, Sebastian.
Thank you, Dave, for the kind introduction. Hello, everyone. It's very welcome to our webinar. So my name is Sebastian Lalouvet, and I am a team manager in the Cell Line Development Unit at Salonic Group. Thanks a lot for joining us today. It's really my pleasure to guide you through today's webinar. Here is the outline of the topics I would like to cover today. First, I will briefly introduce Salonic and the cell expression platform that we use for cell line development. That we use especially focusing on the GEG cell line for perfusion manufacturing. And I will then present some case studies that showcase the advantages of using cell perfusion processes in human cell lines for the production of difficult to express proteins. Finally, I would like to highlight a novel strategy that we have developed for early material supply using Salonic's GEG perfusion platform. So Salonic is a biologics CDMO, so meaning contract development and manufacturing organization, specialized in mammalian cell line. We offer very comprehensive GMP development and manufacturing service for biologics, including new biological entities and biosimilar, as well as ATMPs. And we serve clients across the globe. Indeed, we have a very strong track record and a global clientele in Europe, as well as in US and in Asia. And we have been serving clients all over the world for more than 30 years. Salonic is a fully privately owned company with over 350 employees. We are headquartered in Basel, Switzerland, where we also have our development unit and a small GMP facility. We have our second site in Heidelberg, Germany, where we have our main GMP facility with more than 200 employees. So what do we offer in terms of cell line development? We have two very standardized cell line development platforms, and both are fully customizable for your specific needs. These are the Evolution and the GEG expression platform. Both cell lines are fully GMP compliant, and the cells have been adapted to serum-free media. The biopharmaceuticals which have been derived from these two cell lines have been used in clinical trials in Europe, as well as in US and in Asia. Evolution is based on the non-genetically modified CHO-K1 cell line. It has been proven to be very robust with very high productivity, great stability, as well as excellent scalability. It is mostly used in a fed batch processes, and it comes with a very simplified licensing model. The GEG cell line is a human cell line. It's a derivative from the K562 human cell line. The term GEX or GEX comes from Glyco Express. And this cell line has been developed by a company named Glycotop. It is mainly used in a perfusion process, and it is well established for the expression of difficult to produce protein. For the rest of this webinar, I will focus on the GEG perfusion platform and its advantages. So what are the main advantages of using a human cell line? Well, it's a human cell line, so this means it's ideal for the manufacturing of human therapeutics. What does it imply? As you may know, some quality attributes are really critical for pharmacokinetics and immunogenicity of biopharmaceuticals. Among the CQA for biopharmaceutical is the protein glycosylation. Indeed, most of the difficult to express proteins, such as blood factors, hormones, are hyperglycosylated, and their bioactivity and biostability are highly dependent on their glycan profile. Moreover, it exists a great diversity of end glycans. Some of these glycans are unique to the human cell line. That's, for example, the case of alpha-1,6 sialylation or branching and glycans. And these glycans are completely absent in non-human cells. It is known that these glycosylation are critical for the plasma half-life, the solubility, and the activity of some biopharmaceuticals. On the contrary, some glycosylation, such as the alpha-gal, are completely absent in the human cell line, but present in non-human cell lines, such as CHO cell line. And it is known that humans have circulated autoantibodies directed against this modification that might lead to immunogenic response once administered to patients. For this reason, the human cell line is particularly suitable for difficult to express proteins such as hyperglycosylated protein or unstable protein. The second advantage of the GEG cell line is that it's a very robust cell line and perfectly adapted for biologics manufacturing. So what are the key features of this cell line? It has a great doubling time, around 24 hours. It can achieve very high cell density, up to 50 million of cells per milliliter. It has also been proven to be resistant to shear stress and very stable in terms of productivity. Moreover, we have several GEG cell lines available in case you are looking for some particular glycosylation pattern, such as low fucosylated biotherapeutics. As stated before, this cell line has been adapted to serum-free media, and it is fully GMP compliant. And finally, the third great advantage of the GEG cell line is that it is exclusively used in perfusion processing. So what is the perfusion processing? It exists different processes for the manufacturing. The main ones are the batch manufacturing, the fed batch, and the perfusion process. In terms of the batch process, what you do is that in your vessels, you cultivate the cells until they die without any feeding or any addition of nutrients. And you harvest your product at the end of the process. In the fed batch process, this process is the most commonly used, especially with CHO cell line. What you do is that you have a very discontinuous feeding with nutrients. This will allow to tightly control the metabolism and the viability of the cells, and you will harvest the product at the end of the fed batch process. On the contrary, the perfusion process is very different in a way that you have a continuous feeding, so you will provide new nutrients and you will renew the nutrients all over the experiment, and as well as you will remove the byproduct because you will harvest continuously the byproduct as well as the products. And this is very interesting in case of unstable biopharmaceuticals. As you harvest continuously, you can stabilize better the molecules compared to a final harvest in the fed batch process. At Celonic, we have more than 20 years of experience in perfusion. We have done more than 130 GMP perfusion batches, and we are running perfusion bioreactor from 10-liter scales to 1,000-liter scale. These perfusion batches have been done with very different type of molecules, classical monoclonal antibodies, IgG, IgA, IgM, but also with more complex glycoproteins such as blood factors and hormones or antibody-based fusion proteins. It has been applied to human cell line as well as CHO cell line. So now I will present you 4 different case studies which demonstrate the advantages of the perfusion strategy compared to the fed batch strategy and the advantage of the GEX cells compared to the CHO cells. So in this first example, I will show you how we can decrease the cost of goods by extending the duration of a perfusion process. In general, a perfusion process is done for 30 days. But if we can prolong and extend the duration of this process, we can increase the volumetric productivity. And in this case, it's quite interesting to do it with difficult to express protein. So what we did is that we used our GEX cell line to produce a monoclonal antibody, and we perform a perfusion run over 45 days. And as you can see in this data, we were able to maintain a high cell density all over the 45 days of the experiment. We could achieve a very high viability above 80% after 45 days, and we could also maintain the productivity all over the run during 45 days. So just by prolonging this experiment, we could increase the volumetric productivity as well as reduce the cost of goods, which is very critical when you are working with difficult to produce protein. In the second case study, here is to demonstrate and compare side by side the fed batch versus the perfusion process. So what we did is that we use a GEX cell line expressing a monoclonal antibody, and we compare with the same type of vessels, a 200-liter bioreactor, either a fed batch, which was performed for 14 days, so we did 2 runs of fed batch, or 28 days perfusion. And we look at the productivity which was observed in this comparison. So of course, as you can observe, the media consumption was much higher in the perfusion mode, as we are continuously feeding the cells with fresh media. But at the same time, we could achieve higher product quantity with 3 times higher product quantity in the perfusion mode compared to the fed batch mode, and 6 times higher volumetric productivity compared to the fed batch, demonstrated that we could harvest much higher quantity in the perfusion mode compared to 2 fed batch runs. In this third case study, we look at the quality of the product in our perfusion run. And for this, we look at the scalability between the 1-liter R&D bioreactor to a 200-liter GMP bioreactor. Here, what we analyzed was the post-translational modification, which is the glycosylation, and look at the quality and the relative abundance of the end glycans during a perfusion run for a monoclonal antibody. And we tested this monoclonal antibody in 2 different cell types, one which was a low fucosylated and a second one which was a high fucose. And as you can see, we could maintain all the glycosylation patterns such as fucosylation, sialylation, and galactosylation all over the different scale in the low fucose cell line as well as in the high fucose cell line, demonstrating that we can really reproduce high quality between the different batches at the different size of bioreactor in our 2 cell line. Finally, in this last case study, we look at the expression of a difficult to express protein. This was an antibody-based fusion protein, and we were unable to produce this protein in the fed batch process. Several CHO cell lines have been tested without any success. So what we did is that we performed perfusion process with the same CHO cell line, as well as we tested the expression in the human GEX cell line. So we did 30 days of perfusion with the CHO cell line, and what we observed is that we could get high productivity for the difficult to express protein in the CHO cell line compared to the fed batch where nothing was produced. But more strikingly, with the GEG cell line, we could drastically improve the productivity. 5 times higher productivity was observed in the GEG cells compared to the CHO cells for this difficult to express protein, demonstrating again that the human cell line is very suitable for difficult to express protein. So to sum up this first part, as I mentioned before, the GEG perfusion is really critical in order to have high productivity of difficult to express protein, in order to maintain the quality of the product all over the perfusion run. And it's also important to stabilize difficult to express protein for example. We are, of course, always innovating around the GEG perfusion platform and we have mainly 3 parts we are improving. The first one is always, of course, to lower the cost of goods. As we are using a lot of media in the perfusion run, we are always testing new media and new media composition in order to decrease the cost of goods, but also to increase the titers. We are also working on fastening the timeline for the cell line development in the GEG cell line, because the GEG cell line has currently a higher doubling time compared to the CHO cell line. And finally, the third axis is the early material generation. Indeed, it will be very interesting to provide a service to people who are interested in either doing feasibility in the GEG cell line for difficult to express protein, or just to do as well preclinical studies with early material we can provide in GEG cells. So this axis I will develop in the coming slide. So the early material supply in GEG human cell line is supposed to be just a scale-up of a standard transfection protocol from stable transfection to transient expression in GEG perfusion cell line. But there are some specific requirements. One of the main requirements is that we are bound to some specific transfection method. Compared to the CHO cells, which are easily transfectable with liposuction or lipofectant reagents, the GEG cell line is bound to the electroporation method for the transfection. The second challenge is the fact that we are in a very different scale between a stable cell line, where usually we are electroporating millions or 10 millions of cells, and at the transient scale, where we would like to transfect up to billions of cells, 10 billions of cells. And third, as stated before, the GEG is fully bound to the perfusion process. And so we have to adapt the post-electroporation culture to the perfusion process. And there was, when we started, a high discrepancy between the volume we were using for the post-electroporation culture and the volume we were doing for the transfection electroporation. Indeed, our benchmark in terms of culture was our 3-liter bioreactor in a perfusion mode. On the contrary, the transfection was mainly applied in small cuvettes with a 10 milliliter culture. So what we decided is to develop technologies in order to have a continuum between the 10 milliliter scale and 3-liter scale. For this, we had to scale down our perfusion model to a 10 milliliter scale using either Amber 15 or the shake flask in a perfusion mimic mode. And we had to scale up our transfection electroporation of the GEG cells to 10-liter scale. So now I will present to you the results we have obtained for the scale down of the GEG perfusion culture. So as a benchmark, we used our 3-liter bioreactor perfusion run. It was a GEG cell line expressing a monoclonal antibody in a stable manner. And you can see the data we obtained in terms of viable cell density, viability, and titer. In terms of viable cell density, we achieve a very high cell density with this clone, with up to 40 million of cells that could be observed. We have a very good viability over the 35 days of perfusion run with more than 70% viability at the end of the run. And we achieve very high titer with more than 200 milligrams per liter per day in this 3-liter bioreactor run. As you can see, there was a decrease in terms of the titer over the time. This is mainly due to the fact that the clone we used for this perfusion run was not fully characterized, and we did not know the stability of the products and the cell line in this setup. So now what we did is that we implemented a perfusion mimic mode with the Amber 15 at the 10 milliliter scale. So on the right panel, you can see the results we got with the Amber 15, and I compare side by side the data which have been obtained with the 3-liter bioreactor, which are highlighted in gray color. And as you can see, in terms of the titer in the Amber 15 perfusion mimic mode, we obtained very similar titer around 200 milligrams per liter per day. The viability was very similar between the bioreactor and the Amber 15. The only variation we observed between the two setups was the viable cell density. We achieved higher viable cell density in the bioreactor compared to the Amber, most probably due to the fact that we use a sedimentation mode in the Amber, and this could lead to a decrease or at least a slowdown in terms of the doubling of the cells. But overall, we had a very good transfer between the 3-liter bioreactor data and the Amber 15 perfusion mimic data. We did exactly the same in the shake flask. So for the shake flask, we performed the same experiment over 7 days. We mimic the perfusion using centrifugation steps. And as you can observe, in the shake flask, we obtain a very similar pattern compared to the 3-liter bioreactor with up to 250 milligrams per liter per day, and VCD and a viability which was very similar between the two. So now, in the next step, we had to scale up our electroporation, and for that we investigated what were the electroporation devices that were able to do a continuum between millions of cells to be transfected and billions of cells to be transfected for transient transfection. So for this purpose, we decided to test the electroporation device from MaxCyte, and this company is expert in terms of cell and gene therapy for the electroporation of T cells, for example, or NK T cells. So we tested one of the devices which has really interesting features. One of the features was the fact that we were able to use it in a static electroporation mode with small or large cuvettes up to 100 millions of cells. The second setup was a discontinuous flow electroporation we could perform with large processing assemblies. And finally, the third feature which was interesting is that this device was fully GMP compliant. So we decided to test this device. The first design of the experiment was to test the expression of a GFP using a GFP expressing plasmid, and we electroporated GEG cells at a small scale in small cuvettes. And we look at the expressibility of the GFP as well as the cell viability. In the left panel, you can see the results we obtained via flow cytometry for the analysis of the GFP expression. So we did this experiment in duplicate with completely independent run, and you have the results we obtained at 24 and 48 hours. And as you can see, the cells that were electroporated completely shifted on the far right in terms of GFP expression, demonstrating that these cells were expressing very high level of GFP, and more than 95% of the cells were positive. More strikingly, we observed the exact same kinetic over this 72 hours period of time in terms of GFP expression, with a slight decrease in the intensity of the GFP at 48 hours as well as 72 hours. And this was exactly replicated in the second experiment. We also look at the cell viability at the small-scale GFP electroporation. And as you can see, the control cells, which have been electroporated with control plasmids, were above 90% of viability after 72 hours. When we look at the viability of the GFP transfected cells, there was a slight decrease which was observed at 48 hours, but cells were able to recover nicely with more than 80% cell viability at 72 hours post-electroporation. We were very confident in the results we obtained, so we did just one run of improvement in the electroporation parameters, and we directly decided to test this method with monoclonal antibody expressing cells. So what we did is that we transfected our cells with monoclonal antibody expressing plasmids, and we tested two different scales of electroporation. A 10 milliliter scale electroporation in small cuvettes, and a 1 liter scale electroporation in a discontinuous flow electroporation. And we look at the expression of the antibody using FACS staining, and as well we look at the cell viability of the cells over the 72 hours. Here again, you can see the results we obtained with flow cytometry, and we did three completely independent experiments. The first one was a 10 milliliter scale antibody transfection. Second one was a duplicate, and the third one was a 1 liter scale transfection with the same plasmid. And you can see on this panel the expressibility of the IgG we observed in the cells at 24 hours as well as 48 hours. And as you can see in this FACS plot, we observe very similar expressibility of the IgG in our cells in a transient manner at 10 milliliter scales between the two replicates as well as the 1 liter scale, demonstrated that there was a very high scalability between the 10 milliliter scales and 1 liter scales. Later, we look at the cell viability of the cells. Again, the control cell had more than 90% of viability after 72 hours post-electroporation. At the 10 milliliter scale in the blue color, you can see the viability of the cells, which was also slightly dropping after 48 hours, but cells were able to recover nicely after 72 hours with more than 80% of cell viability. At the 1 liter scales, we observe a slight difference. The cells had a lower viability, around 65–70%, but still the viability was above 70% after 3 days of post-electroporation. So meaning that the cells were viable and we were able to use these cells to test the transient expression of our antibody. Based on these very encouraging results, we decided to continue the experiment and merge our electroporation scale-up as well as our perfusion mimic mode in a shake flask. So what we did is that we electroporated the GAG cells at a 1 liter scale with our monoclonal antibody expressing plasmid, and we maintained the cells over 7 days in a perfusion mimic mode in the shake flask. And yeah, as you can see, we could observe a doubling of the cells over this time period with a viable cell density that reached more than 10 million of cells per milliliter. And we could observe a nice expression of our antibody in terms of the IgG expression, which was more than 40–50 milligrams per liter per day. So based on all these experiments, we can now offer a service where we can provide early material for in a GEX perfusion setup for any feasibility study and preclinical studies for difficult to express proteins. To sum up all this webinar, I hope I convince you that the GEX human cell line and the perfusion process are highly suitable for difficult to express protein production. I demonstrated that we were able to optimize the GEX perfusion mimic processes for early material generation. We could set up a large-scale GEX electroporation for early material generation, and we are very proud to offer a new service with our GEX perfusion setup for fast-track early material generation. Then I would like to thank you for listening. We are at CellLink, we are always working on the improvement of our development and manufacturing environment in order to deliver the best and tailored biopharmaceutical solution to our clients. And I really would like to thank all the people who have contributed to this work, especially the CLD department, the USB department, as well as people from our GMP facility in Heidelberg. I really thank you for your attention. I hope you find this session very informative. You can always reach out to me with your questions via email or phone, and the session is now open for your questions.
Thank you very much, Sebastian. Very much appreciated. And the audience does have some questions for us. The first one is from Jiho, and they ask, what is Celonic's GMP capacity in perfusion mode?
So in terms of the GMP capacity, so yeah, as I stated in the development department, we have scaled up to 3 liters. In our GMP facility in Heidelberg, we have bioreactors in a perfusion mode from the range of 10 liters to 1,000 liters. And this is our max capacity we have. But knowing that with 1,000 liter bioreactor in a perfusion mode, we can harvest up to 70,000 liters of media. So it's a huge capacity.
Thank you, Sebastian. The second question is from Jennifer, and she was asking, what types of difficult to express proteins have you manufactured with GEX at Celonic? Yeah,
so of course, out of all the classical monoclonal antibodies we have expressed, such as IgG, IgM, IgA, we have a strong experience, especially in the field of the hormones and the blood factors, which are usually hyperglycosylated protein. We also have experience, as I showed to you, in antibody fused proteins, as well as some enzymes. So these are our main experience with difficult to produce proteins.
Thank you, Sebastian. The third question is from Jean. And Jean asks, what clinical phase are the products made in GEX at present time?
So in terms of the clinical phase, so for the GEX cells, so we have more than 40 batches in a perfusion mode which are being performed. And all these products, biopharmaceuticals, have been used in phase 1 as well as phase 2 human clinical trials.
Thank you. The next question is from Hintendra. And the question is, with fed batch versus perfusion technology, what was the specific productivity in perfusion?
Oh, I have to admit that I don't know it by heart. I will need to look more deeply to this question, but I will be more than happy to reply to your question when I have this data.
Fabulous. Thanks, Sebastian. The next question is from Christina, and her question is, when is it proper in development to consider testing GEX and perfusion technology if expression in CHO failed in their first experiment?
Could you just repeat the question? I did not hear the last.
Sure. When is it proper in development to consider testing GEX and perfusion if expression in CHO failed in their first experiment?
I think nowadays with our early material generation, we can perform. I think at very early stage in the development, we can evaluate the potential of the GEX cells and the perfusion mode. So by doing already transient expression of the molecules, we can already look at the expressibility of the molecule of interest and see whether the GEX cells is fitting the requirements in terms of glycosylation or expressibility. So it can be done at very early stage in the development phase.
Great, thank you. The next question is from Jeff. And Jeff asks, if my company chooses to use a GEX cell line for development, what are the commercial requirements associated with GEX?
So the GEX, as with our CHO evolution, are coming with a very simplified licensing model. There are no royalties on the sales of products made with the GEX cells, no licensing fees for products if the manufacturing is, of course, performed at Salonic. It's a very simplified model we would like to offer to our clients for both CHO and GEX cells.
Thank you. And the last question I have is from Richard. And Richard said, you said GEX is a human cell line. Have regulatory agencies accepted use of this cell line for use in humans?
Yes. So this cell line has been or is fully compliant to all the GMP guidelines. Some biopharmaceutical expressing GEX have already been approved by the FDA and EMA for human clinical trials. And all these regulatory authorities have confirmed that this cell line is, which is fully characterized, and it's fitting those regulatory requirements.
Thank you, Sebastian. Very much appreciate your presentation and your answers today. That's all the questions we have at the present time. I want to thank everyone for joining us today. And as a reminder, please complete the survey that will show up on your screen shortly. If you have any additional questions, which we weren't able to cover today, simply send them to GEX, G-E-X, at salonic.com, and the team will provide you an answer or answers to your questions. And we want to thank you again for your participation today. Most appreciated, and we look forward to speaking with you again soon. Have a great day.
Thank you. Have a great day.